Hello, this is. I just want to get us started with our genetics unit with some vocabulary. Now, I've already written them down on here, so you are welcome to pause when necessary when you need to. So the first thing we need to go over is the difference between asexual versus sexual reproduction. In asexual reproduction, there is no division in the chromosomes. There's no mixing of the DNA. So whatever offspring you make, it's a clone. So plants are a really good example of this, where you could take a little snippet of a stem in some plants and root it in dirt, and oftentimes it'll grow in other plants. And they do this naturally, like they can spread runners, and those are clones. Now, sexual reproduction involves a mix of chromosomes with another organism, with another, you know, uh, member of that population, of that species. So the gametes that form that organism would be the sperm and the egg cells. Those are the gametes that then mix. And because those chromosomes are coming from mom and dad, you've got a variation in the offspring that are a mix of the mom and the dad. Now, so the next thing we need to look at would be the somatic cell. And the somatic cell, soma means body. And so the somatic cell are body cells. Now, body cells are going to be diploid. And diploid means that there are two sets of chromosomes. So we all have pairs of chromosomes. And if you have two full sets of chromosomes, then that cell is diploid. So you can remember that from the di in the diploid. And I realized I mistyped that. There should be an L there, diploid. Now, a gamete is a sex cell. It would be either the egg, and the fancy term for that would be an ova, or a sperm cell. Now, those cells are haploid. They only have one set of chromosome. Now, for us, we would represent that as 1N for any organism. For us, that means there's 23 chromosomes, 1 through 23. So a diploid cell would have 46, but the sperm cell and the egg cells only have 23. Now, the last pair for us and for many mammals would be the sex chromosomes, and they determine the gender. So that's the 23rd chromosome. Now, for us, the X chromosome is a normal shape, typical chromosome. So this would be the X, and you can see why they call it the X. The Y chromosome is actually truncated. It's actually missing a little bit of the chromosome. So when they saw that under the microscope, they originally called it Y. Now, the Y chromosome is actually missing some of the genes, and that's why in sex-linked genes, if they're down here on the X chromosome, then that means we wouldn't even see it on the Y, like the Y is not even carrying it, and that's why it's significant. So non-sex chromosomes are called autosomes. So they're on chromosomes 1 through 22. And so the difference between sex-linked genes are going to be found on that X chromosome. Autosomal genes are going to be found on chromosomes 1 through 22. Now, when we're seeing chromosomes, we have other vocabulary for the chromosomes. For instance, we have locus or loci for plural. So location of gene is simply where it occurs on the chromosome. For instance, we could theoretically point to that little orange banding and say within that orange banding, that's the loci, that's the locus of our hair color gene. And maybe on one chromosome, you have a gene for black hair, and then the other chromosome, you have the gene for hair color. Now, the chromosomes that stain the same way, so notice they've got purple and blue and orange and another blue and green. If they stain the same way and they're the same length, then they are called homologous chromosomes. They are coding for the same alleles and same genes. So they have the same genes, they are the same size, and they stain the same way. And the two homologous chromosomes themselves, if they're duplicated, they will have two sister chromatids. So here's one sister chromatid, here's the other sister chromatid. And when we align them together, we would have a karyotype. And the karyotype is where we arrange all of the homologous pairs. So notice we've got 1, 2, 3, 4, all the way up to chromosome 23. And here's our X and here's our Y. And again, notice with our Y, here's our little Y right there, and it's very, very short. Now, the sister chromatids are hard to see in these diagrams, but what you end up having here would be one chromosome, sister chromatids, and then here would be its homologous chromosome with its sister chromatids. Now, on one chromosome, they might carry the gene for blonde hair, blonde. Over here, in the same locus, you would have the gene for black hair, or brown, or, you know, whatever hair color you want to put in there. And so that's how we're looking at these chromosomes. Now, they're arranged according to their size. So notice number 1 is the longest one, and they get shorter and shorter and shorter as they go on from 19 to 23. And of course, 23 is the odd one out. We got one long and one short. Now, so this gets us to the genes. If we have black hair and blonde hair, we would call those alleles. We might say in dogs, you might have an allele that has brown spots and an allele that has black spots. So an allele is a variation of a gene. It's coding for a different protein product. One receptor for, like one protein for a brown spot, one different protein for black spots. Now, if an organism has both alleles the same, we would call it homozygous. They are the same alleles. Now, that's very similar to pure breeding. Pure breeding can only produce one type of offspring. So pure breeding is an older word when they didn't understand necessarily what was happening. Pure breeding means that you're only going to produce one type. Well, that means they're homozygous. They're only giving their offspring one type of allele. Either they're only giving them dominant or they're only giving them recessive. So both of these are homozygous. Heterozygous means that you have different alleles. So a big A and a little a. So a parent might give their child a brown spot allele or a black spot allele. So since it's Christmas time, and if you've had me before in class, I always like to use Rudolph. Now we can consider two different types of genes. So we could say Rudolph could potentially have a black nose or a red nose. Rudolph could also potentially have a gene for cannot fly versus a gene that can fly. Now in reindeer, if those are the possible alleles, then what would Rudolph be? Well, Rudolph has a red nose, so we better have two little B's for a red nose. That's the only way you can get a red nose if it's recessive. A recessive allele means that it's only expressed when the dominant one is not there. Now, that doesn't mean it's not strong enough, and we'll talk about like mechanistically what's happening. It simply means that if the dominant allele is there, it's going to be promoted preferably over the recessive gene. And then for Rudolph, because he can fly, he would be little f, little f. So in that case, he has to be homozygous recessive for all traits. So dominant means that it's going to be expressed even if there's only one allele, that gene kind of takes over if it's a completely dominant type of gene. And a recessive means that you're only getting that trait if it's homozygous. Now, if we keep going, the genotype is the arrangement or combination of the alleles. So if I said homozygous, so big A, or big A, or little a, that is a genotype. That's the arrangement. Versus the phenotype, which is the physical observation. So you could be heterozygous, but we're really only seeing the result of that big A gene. So the phenotype is what we can physically observe. So you may have brown hair, that's your phenotype. Your genotype may be heterozygous. You're carrying the blonde hair gene. Now, independent sorting will come about in meiosis, where the chromosomes line up with their homologous pairs in a random process. So let's apply what we've talked about here. In meiosis, during metaphase 1, the chromosomes are going to line up. Now, they're going to line up with their homologous pair. Now, if we take the example of Rudolph, and let me use my red pen. So let's say in this top chromosome, we have one gene, and then in our bottom chromosome in a different spot, sorry, let me put it down here. In our bottom chromosome, let's say we have a different gene. So let's go ahead and label those. So this might be the nose gene, and this might be the flying. Now think about this from a probability standpoint. We have a couple of different ways that this could arrange themselves. On the top, we might have the red nose on both of these chromatids, and then on this one we might have the black nose. Down at the bottom, we might have can fly. Over here, we might have no fly. However, think about this from a probability standpoint. We could find it arranged differently. We could have, let's keep the top one the same. We could have a red nose on these sister chromatids and the black nose on the other one, just like we had before. But let's say we switch these two. They could line up differently. This one could be no fly. This one could be can fly. Now, this is no different if I gave you 4 cards and I said, okay, you've got hearts, diamonds, clubs, and spades. How could they assort themselves if I said the red cards had to be on the top? And so you might say, okay, well, I have a heart and then a diamond and then a club and then the spades, right? But you could say, well, that's one arrangement, but maybe down here, I could also say, well, the spades could be that way, and then the club could be the other way. So how they pair up is random. And that we'll see that again and again. So keep that in mind. How they line up during meiosis is going to dictate how they sort to their cells. So we're going to talk about meiosis tomorrow and compare them. So hopefully this gets you started.